Good afternoon, everyone. Welcome to our webinar, Aggressive Breast Cancers in Black Women. I'm Dominique Bethia, the Outreach Manager at SHARE. Before our presentation begins, I would like to tell you a little bit more about our organization. SHARE is a nonprofit that has been helping people through breast, ovarian, and uterine cancer for the last 45 years by offering the support of those who have been there. SHARE provides many services, including a helpline, support groups, and educational programs. All services that we have are free of charge to participants. For more information, please go to our website, sharecancersupport.org. Just a little bit of housekeeping before we get started. All participants will be muted for the presentation. When Dr. Ambrosone and Dr. Yao finish presenting, we will begin our Q&A discussion. You are welcome to submit your questions during the presentation through the Q&A button that you will see at the bottom of your screen. The chat section will be disabled. When you're asking questions, remember, the presenter is unable to give specific medical advice. So please try to keep your questions general in nature. The webinar is being recorded and it will be available on the SHARE website very soon. Now, without further ado, I would like to introduce our two speakers. First, we have Dr. Christine Ambrosone, PhD. She's the Senior Vice President for Population Sciences and Chair of the Department of Cancer Prevention and Control at Roswell Park Comprehensive Cancer Center. She is a molecular epidemiologist whose research focuses on both the causes of breast cancer and factors that influence cancer treatment outcomes. Much of her current work focuses on breast cancer disparities, and she previously led a large multi-center study to identify genetic and non-genetic risk factors for aggressive breast cancer in Black women. She is currently researching molecular mechanisms for estrogen receptor negative breast cancer and its outcomes. Dr. Ambrosone has served as a standing member of the NCI study section related to cancer epidemiology and was a member of the board of scientific advisors to the director of the NCI. To date, she has more than 380 publications in peer-reviewed journals. And our other presenter, Dr. Song Yao, is a professor of oncology and director of molecular epidemiology at the Roswell Park Comprehensive Cancer Center. He is a molecular and genetic epidemiologist in training, by training. He has been working in the research area of cancer health disparities for more than a decade. He is a leading multiple NIH-funded projects with a major focus to understand what type of unique biological changes in breast cancer cells in Black women that have made the disease more aggressive and deadly in this population. He has published more than 100 peer-reviewed papers, a majority of which are on the topic of cancer health disparities, and many in prestigious journals in the field. I now present both of them to you. Christine?
Thank you. We're very happy to be joining you and to be doing this presentation. And now I'm going to share my screen. Is this good?
Yes, good.
Okay, great. All righty. So,
Can you go to full screen mode?
oh, I thought I was in it. Oh, Christine.
It's at the lower right corner.
Well, I know I've got, the slideshow, it's there. And I've got that on, but there we go.
There you go. Good.
Okay. All right. So today I'm going to talk about the work that we've been doing to try to understand breast cancer in Black or African American women. Historically, when I first got interested in breast cancer, the thinking was, and the knowledge was, that white women had higher rates of breast cancer than Black women. But as I'll show you in the next slide, these rates are changing. However, more Black women have been dying from breast cancer than other groups in the US. And with time, these rates have changed. So this is breast cancer incidence, and the lines are, this is white women, Black women, Asian Pacific Islander, et cetera, and Hispanic. And here you can see that the incidence rates start out higher for white women than Black women up until about 2005. Part of this is them changing is because we found out that hormone replacement therapy increased risk of ER-positive breast cancer. A lot of women stopped, and so these rates came down. But for Black women, the rates have not gone down and have continued to increase. Now, if we look over here at deaths, we see a different picture. This is really from 1975 to 2012, these data. And you can see mortality is much higher for Black women than it is for white women and for the other groups. So that part of this reason for higher mortality may be due to less access to screening and this later stage of diagnosis, and less access to the best possible treatments. When I first started studying this, I lived in Arkansas, and it was a real eye-opener because in the Mississippi Delta region, there's no buses and public transportation. Hospitals are far away. So to even go for mammogram and to get free to be able to do that is very difficult. And then if you are prescribed radiation therapy, getting back and forth to the hospital every day. So socioeconomic status or other factors could result in higher mortality because of later stage of disease and also inadequate treatment. But more and more we have found that Black women are more often diagnosed with tumors that are more aggressive and have worse outcomes. So breast cancers can express receptors for estrogen, progesterone, and HER2. And estrogen is a main driver because that's what makes the cell replicate and makes the tumors grow. And some cancers, some treatments for breast cancer block estrogen. And so they are easier to treat. However, many of the tumors in some Black women are negative for estrogen receptor, progesterone receptor, and HER2, and these are called triple negative breast cancers. Up until recently, there have been no targeted therapies available for triple negative breast cancer, such as tamoxifen and aromatase inhibitors. So we think that that may be part of the reason where we see higher mortality. And this is distributions of breast cancer subtypes in the US. Red is everybody together, and green is non-Hispanic whites, and the blue here is non-Hispanic Blacks, and then Hispanics and Asian Pacific Islanders. So this is hormone receptor positive, HER2 negative, and a little lower rates in Black women. But this is triple negative breast cancer. And here you can see it's almost twice as common in Black women than it is in white women. So these are inherently usually higher grade tumors and harder to treat. And there's no targeted therapies for it yet, though there are some studies in the field now looking at use of other treatments. So why are these aggressive cancers more common in Black women? We started, when I lived in Arkansas, I started, this was 20-some years ago, 25 years ago, actually, trying to understand this and work on this question. So a couple of years later, I moved to Mount Sinai in New York and started the Women's Circle of Health study. And we started in Manhattan and then worked with a colleague at Cancer Institute in New Jersey and expanded the study to New Jersey. Many years, different funding sources, and this study is just about winding down now. So what do we know about risk factors for aggressive breast cancer? Well, in the past, what we knew about breast cancer came primarily from studies of white women, and most of them had ER-positive disease. So when we think about what the causes are of breast cancer, that's mostly relevant for ER-positive breast cancer. And at the time, it wasn't really known if risk factors might differ by estrogen receptor status, or if there were differing risk profiles for Black women. We do know from those studies of primarily white women, there's increased risk with family history of breast cancer, radiation exposure. Hiroshima, it was the girls who were about 12 years old with developing breasts who got breast cancer later rather than adults. Hormone replacement therapy, alcohol consumption, overweight in postmenopausal women, and sedentary behavior, low physical activity. Key risk factors, though, are reproductive risk factors. So there's lower risk with later age of menarche. So if girls have their first periods later, they'll have lower risk of breast cancer. Having children, we thought reduced risk, so we call this parity, as well as earlier age at first birth and early age of menopause. So anything that kind of shortens that period of time that the breast is exposed to estrogens and hormones results in lower risk. But in 2008, this paper came out that just knocked me out. So luminal A breast cancer it's what's estrogen receptor positive, HER2 negative, and basal-like triple negative breast cancer. So the Carolina breast cancer study is in North Carolina. It was led by Bob Milliken at the time, and it enrolled hundreds of white and Black women and got a lot of information from them, as well as genomic DNA and tumor tissues. And so here, I'm going to show you odds ratios. So we compared women with breast cancer to women who didn't have breast cancer. And we compared those who had no children to those who had children. So an odds ratio is the ratio of risk with the exposure over that without. So if a ratio is 1-to-1, there's no risk. So we work from there. And here, an odds ratio of 0.7 is reduced risk by like 30%. So having children, doesn't matter how many, is associated with reduced risk of ER positive. But here, having children is associated with increased risk. And this is almost a twofold. So 1.9 times more likely to get breast cancer, ER negative breast cancer, triple negative breast cancer, if they had 3 or more children. This, I knew Bob Milliken pretty well, and I called him and I said, Bob, this is amazing. Why isn't this in New England Journal of Medicine? Why isn't this on the front page of the New York Times? This is big news that there's such difference. And he was a diligent scientist, and he liked this journal because they let you publish as many pages as you want, so he can tell the whole story and explain it all. But what was really incredible was looking at whether or not these women breastfed. So here we have having 1 or 2 children and not breastfeeding, having and then 3+, and then having 1 to 2 in breastfeeding or 3+ in breastfeeding. Again, we see breastfeeding doesn't change at all the effect of having children. We see reduced risk no matter how many children and whether or not they breastfed. Here again, we see the increased risk with having children, but if women breastfed, that increased risk almost totally goes away. And that was what just knocked my socks off. I thought this is huge. If we could reduce risk of aggressive breast cancer just by encouraging women to breastfeed, it could make a huge impact on the toll that breast cancer takes. So that paper really didn't receive any publicity. People weren't aware. I gave a talk to a very large audience at a really big meeting and asked how many people knew this, and almost no hands went up in the audience. But Black women were more likely to have children at a younger age, to have more children, and less likely to breastfeed. So I was thinking this could have a huge impact on poor prognosis breast cancer in Black women, as well as white women, through breastfeeding. So there were fairly small studies afterwards looking at this, including the Women's Circle of Health study that I showed, the Black Women's Health study, which is a very large prospective cohort led by Julie Palmer, seeing the same findings, but people still weren't thinking about this. Risk factors for breast cancer, having children reduces risk, and not this popped out that it increases risk of ER negative, but breastfeeding can take that away. So clearly we needed a big study to pool data and to confirm those associations and get the word out. So this is Bob Milliken, who became deceased shortly after we started the study. It was heartbreaking. And Julie Palmer and I, and we were at a meeting that NCI held on breast cancer in Black women. They were talking to us about pooling our data to look at genetic differences. And Bob and Julie and I got together out in the hallway and we said, let's pool our data and look at having children and not breastfeeding in ER negative breast cancer. We will have the numbers where we can really make an impact. So we got together, we got Larry Collin now with the multi-ethnic cohort involved, and that's what we did. We got a program project grant. We called it the AMBER Consortium, Epidemiology, Breast Cancer Subtypes in African American Women. And with that, we had almost 4,000 women with breast cancer and more than 14,000 controls to be able to compare. So here's another way of looking at data. These are odds ratios. This is 1, no association. If it's below 1, there's reduced risk. If it's above 1, there's increased risk. And here we looked at having children or not for ER positive. No, they all had children, but whether or not they breastfed and ER positive and ER negative. So this is ER positive breast cancer. And if they breastfed, they had reduced risk of ER positive breast cancer, but they still had reduced risk even if they didn't. And that's like the same as the Milliken data. It didn't really make that much difference. But for ER negative breast cancer, if they did not breastfeed, we see this big increased risk, almost twofold. And if they breastfeed, the risk is much lower. We still see this increase here for having 4 or more children and breastfeeding, but to a great extent, breastfeeding reduced that risk. So in summary, for ER negative breast cancer, the highest risk is for women who have children and didn't breastfeed. And as in the Milliken study, there was reduced risk of ER positive disease with having children regardless of breastfeeding. So I want to segue a little bit because at the same time that we were working on this, or actually before we were working on this, we used to get together every couple of months with some of the folks in immunology. We had a hotel connected to Roswell, and we go over there sometimes and have a drink and just talk about what we were doing in our research interests. And I was talking about more aggressive breast cancer in African American women and more poor prognosis prostate cancer in Black men. And Cornelia Duncy, immunologist, said, well, of course, there's very different immune response in people with African ancestry than those of European ancestry. And we just took off with this and had a lot of discussions, a lot of reading, a lot of talking, and developed this hypothesis that over millennia, like millions and millions of years, differences develop between populations from different continents. So we know that mankind began and evolved in sub-Saharan Africa, and to protect from endemic infectious disease, they developed very robust immune inflammatory responses. And one example is there's a gene, Duffy antigen receptor, that there's a variant that's only present in women of African ancestry. And this protects from malaria. So a lot of these differences in immune response, they were there for a purpose. But with migration over millennia, again, out of Africa into Europe, across to Asia, the need for this robust immune response was not so great. And gradually, again, over millennia, these variants that encoded for these differences went away. So the studying of parity in breast cancer and ER-negative was Project 1, but Project 4 in the Amber Consortium was trying to investigate this concept of immune response, and also, which Song will talk to you about also, and I don't have it written up here, but another, it's thought that sub-Saharan Africans had dark skin, more melanin to protect the skin, right? And with, again, migration over millennia to Europe, you lose that. And with that, well, Song's going to tell the vitamin D story. I'm not going to go there. It'll take up too much time. So I'll let Song take over here because he's done a lot of work. He was the leader of this project, looking at ancestral differences in systemic immune responses. And then he's going to talk about vitamin D.
Yeah, thank you very much, Christine. Wonderful talk. And always amazed about this line of research you first came up with. So, like you said, we were very interested in looking at how the human evolution have left a huge footprint in the immunity in modern populations. And here are 3 papers that came out a few years ago, show exactly just that. So they look at different immune cells collected from different population of ancestral background, and they compare them, and they show consistently that there are remarkable differences in immune response between those different populations. And most of the times they can attribute those differences back to the genetics, and those genetics have been shown to subject to a selection during human evolution. So exactly what we have been hypothesizing. Okay, good. So in our own study, in the Amber Consortium, where we measured 14 chemokines and cytokines from women of European and African ancestry, and we compare the levels between those 2 groups in those chemokine cytokine levels, which are markers people usually use as a way to measure host immunity. And as you can see on the figure on the right, there are significant differences between those 2 groups in those immune markers. And can you go to the next slide? And some of those differences we have figured out are attributed to environmental or lifestyle factors, like people like obesity, like smoking, those kind of environmental factors that certainly have an impact on our immunity. But then after we have controlled for all those factors, we make sure the differences we saw are no longer related with those environmental lifestyle factors. We still see quite dramatic differences in 4 markers. That's where we started to dive into genetics. And we did some genetic analysis. And we started with an agnostic approach. So that means we did not have any specific hypothesis. We just look at the whole human genome and do a screening across the whole genome. And we found a signal in this shown in this very high peak on the figure to the right. You can see there, that means that this region in the human genome is related with such differences in the cytokine levels. And then we went into that region and identify the exact genetic genes responsible for such difference is the Duffy-Now allele. So exactly as what Christine had hypothesized. And it was really wonderful that we started with the hypothesis and then we came back to it to find what exactly what we had thought about. So now back to the question, could those immune differences we have found between Black and white women or Black or white population in general, could that have an impact on the aggressive cancer that we have commonly observed in the literature? So we thus started a study where we use tumor samples collected from a large population of Black and white patients. So this is called the WCHS study Christine started. And we have a pathologist, Sir Corey, helped us to evaluate the tumor infiltrating lymphocytes, which are basically immune cells that got into the tumor regions and trying to eliminate the tumor cells there, because tumor cells can be recognized by immune cells as foreign, and thus there will be a strong immune response and effort to eliminate those tumor cells. And we found that, again, we saw Black patients tend to have a stronger immune cell presence in their tumor than white patients. And that's true for different breast cancer subtypes. And then we next asked whether the immune cells are really, the presence of immune cells in tumor regions are associated with breast cancer survival. And because this has been shown repeatedly in populations of white, but we didn't know at that time in Black patients. So we did this analysis only in Black breast cancer patients, and we saw exactly the same thing, that the stronger presence of immune cells in breast tumor are associated with better survival. So then the question became quite interesting, why Black patients have stronger immune cell presence, why they still have a poor outcome. Then we did another study where we delved a little bit deeper into the immune cells, because there are many types of immune cells. And those immune cells can also have different different status, different activities, depending on the molecular changes. So we looked at a number of molecular markers on the surface of the tumor cells. Those are called inhibitory receptors. And those receptors can mediate a phenotype called exhaustion. So when immune cells express those molecular markers, then the immune cells become exhausted in a way, and they are not very functional, and they don't have a robust anti-tumor activities anymore. So we developed a molecular signature, we call it EXCD8 R signature, which is really a ratio between the exhausted and then the cytotoxic, which is the active immune cells. And we found again that this signature is very strong in Black patients and in white patients. That means more Black patients are more likely to have those exhausted immune phenotypes. Can you go to the next slide? And we associate the T-cell exhaustion phenotype in relation to patient survival, and we had hypothesized that patients with more exhausted immune cells are more likely to do poor after breast cancer diagnosis. And if you look at the bar graph shown at the bottom, the red color means that those patients with tumor that are more likely to have exhausted immune cells, the ratio is much, much higher in Black patients than in white patients. And this is true in our own data set and also in another data set, TCGA from NCI. And so we put together, based on those findings and other findings from our group, we put together a new grant and to expand this line of research to do a more comprehensive systematic analysis on the immune phenotypes in breast tumors in Black and white patients, and also working with immunologist Scott Abrams here. And this grant is funded, and we started a new study based on in collaboration with New York Cancer Registry, and to enroll Black white patients into this study. So this study is currently ongoing. And we are very excited to have started enroll many patients into this study. But then at the same time, we are also interested in another aspect of the research is how we can target the exhausted immune phenotypes we have seen more often in Black patients. There is exactly one new cancer treatment. Many of you probably have heard of cancer immunotherapy. And one type of the cancer immunotherapy is called immune checkpoint inhibitors. So those checkpoint inhibitors as shown on the in the cartoon on the left, those immune checkpoint inhibitors target exactly the inhibitory receptors which prevent the immune cells, our own immune cells, from attacking the tumor cells. So once that inhibition signal is removed by the treatment, then the immune cells can kill the cancer cells and can lead to improved survival. And this has been a very big deal in the last decade or so in the field of cancer therapy, and have changed the prognosis and outcome for many cancer patients who, especially those with advanced cancer before. And as shown in the figure on the right, there has been a very large clinical trial programs ongoing, thousands of trials ongoing on to find the to identify new targets for cancer immunotherapy and to try different combinations of those treatment, immunotherapy or with chemotherapy, to improve patient outcomes. So we are wondering, those apparently leads us to hypothesize whether the differences we have seen between Black and white patients in their immune profiles can lead to differences in the response to cancer immunotherapy. When we went to the literature, really, there are not many studies with a large proportion of African-American patients enrolled, especially in those clinical trials. For many reasons, the participation of minority population in clinical trials is quite low. So we don't have really good data. So that's where we partnered together with a team from University of Rochester, also in upstate New York, here led by Dr. Charles Kamen and Gary Morrow. We two teams combined forces and put together a large study where we call DIRECT. It stands for Disparities in Results of Immune Checkpoint Inhibitor Treatment. Christine coined this name, very nice. And we designed study logo here. And this was designed as a comprehensive study. We are looking at a number of different aspects that may be related with the differences in patients' experience with immunotherapy. So not just biology differences, but also the social economic differences and the patient access to healthcare. So social determinants of health. So this study we just launched, and hopefully we will start to patient enrollment very soon. And this study will be conducted through the large group called NCORP, NCI Community Oncology Research Program, which is a national-wide network. And as you can see here on this map, there are also sites across the nation, and we are going to enroll patients from this excellent infrastructure. And so I'm now going to switch the topic a little bit, as Christine had hinted earlier. Vitamin D is another of my favorite topics, and I've been working on this for a number of years since my PhD training with Christine. And so, as Christine had mentioned earlier, skin pigmentation has a lot to do with how we synthesize vitamin D, because vitamin D is quite low in most of the food items we consume, and we obtain vitamin D mostly from skin synthesis. But the higher skin pigmentation, a darker skin, could compromise that vitamin D synthesis and cause a vitamin D deficiency. So it's shown here in this figure. You can see the skin pigmentation, the skin tone varies widely across different individuals. It's not just black and white, but you can see a whole range of different skin colors, and this certainly has some impact on the efficiency of how we synthesize vitamin D under sun exposure. And in an earlier study based on the Amber consortium, where you can see where we measured vitamin D levels in Black and white women, and we had hypothesized that Black women tend to have much lower vitamin D levels than white women. And if you look at the proportion of vitamin D deficiency, you can see it's 82% in African American, but 40% in European American. So it's more than twofold higher. And then why we think vitamin D could be related with breast cancer is we have done a few studies now. One of the first study, we showed lower vitamin D levels is associated with a poor breast cancer survival. And that's work is in collaboration with Larry Cushy at Kaiser Permanente in Northern California, Oakland. And Christine and Larry had co-PI this excellent breast cancer survival cohort called a pathway study. And we have a very large 4,500 breast cancer patients followed up for an average 10 years now. And we just recently did a new analysis by measuring vitamin D levels in almost 4,000 patients. And we did with a larger sample size, a longer follow-up time. So we look at the association again, and we found exactly what we have found before, that patients with deficient vitamin D levels at the time of diagnosis had a significantly worse survival than those with sufficient vitamin D levels. And then when we look at the vitamin D levels in this study by racial ethnic groups, and as you can see here on the figure on the left, Black patient again had the lowest level of 25 hydroxy vitamin D. And when we look at whether lower vitamin D levels contributes to the poor breast cancer survival between Black and white patients, and the answer is yes, it did contribute to the differences we have seen before between Black and white patients on breast cancer survival. So here is some key takeaway messages from both the presentation Christine and I did. Black women tend to have more aggressive breast cancer and higher death rate from the disease. And breastfeeding lowers the risk of more aggressive breast cancer, especially in women with a higher number of children. And a stronger pro-inflammatory immune response contributes to more aggressive breast cancer in Black women. And this can be targeted by immunotherapy. And we are doing new studies trying to answer this very question. And then maintaining sufficient vitamin D levels may be beneficial after breast cancer diagnosis, particularly for Black women who are known to have lower vitamin D levels. So with that, I'd like to thank you for your attention, and thanks Cher for having Christine and I to be here to share some of our results.
Thank you, Dr. Yao, and thank you, Dr. Ambrosone. Some really interesting things that I never knew. Well, we have quite a few questions. I'm going to start the Q&A. So for the first question, is disparity in treatment protocol caused by lack of Black participation in drug trials? And if so, is it an access issue?
Christine, do you want to take this?
Why don't you, Son?
Sure. Yeah, the participation in clinical trials from minority populations, including Black patients, has been historically low. That has been true for many, many years. And that certainly has a lot to do with the mistrust with the medical system. And also has been a very hot topic in recent years by NCI and by the scientific community, trying to address that very question to improve the enrollment of minority populations into clinical trials, so that will help us to answer the very important question whether the findings from those clinical trials are generalizable to a larger population, to the whole population, or just to a few, or just to white population in most cases. So in the new study we are now launching, we target specifically to enroll a large number of Black patients, so we can compare the two groups. And we are also working with many different parties trying to improve the enrollment of Black patients into trials.
Thank you. Thank you. Dr. Ambrosone, did you want to add to that?
No, thanks. I was very distracted trying to figure out how to stop sharing my screen. We always use WebEx and trying to keep my platforms straight.
Yeah, these days we have a million platforms. So I definitely understand. We have to learn all of them, right? So for our next question, which is something that I've also heard, they're asking, why is it so difficult, or appears to be difficult, to qualify for a clinical trial?
So again, we're not clinicians. I don't treat patients, we just do research, but that can be a problem because specific trials, you have eligibility criteria and exclusion criteria. And, they may want patients with very specific disease characteristics, specific stage of disease, performance status, how healthy they are, how old they are. So there's a lot of criteria, and that may make it harder to get into a trial. But more and more, the cooperative groups who run a lot of these trials are really trying to expand and extend those studies and offer the same, treatment options to everybody.
Okay, great. Thank you. Thank you. So to lead us into our next question, what can we do, as advocates, as cancer survivors, what could we do to help contribute to your research?
Wow. So Bob Milliken, whose picture I showed, who was the PI of the Carolina breast cancer study, decades ago started Project LEAD, where you're familiar with that?
I've actually been to Project LEAD.
Oh, okay. So breast cancer patients, advocates can get really, really trained in the science of breast cancer research. And I've been on panels that have had advocates on them. They contribute so much. And in our research, we actually, this new study that Song showed, we have a community advisory board. They're all advocates. And we took our materials to them, like our brochure, and they said, oh man, that is way too many words. And they really helped us refine it and figure out the best way to get the messages. So I think, if you're offered the opportunity to participate in research, it's so helpful to us. Like right now, you need a lot of people to do research. As Song showed you that vitamin D study, there were like 4,000 people. And the new study that we're starting, we're getting names from the registry. If the people don't, saying no, don't share my name, and contacting patients, asking them to participate. And every failure, it's a shame that because every fewer person that you have, the harder it is to get solid research findings. So you can help our research by saying yes. And being advocates, at the national level as well. That's how the whole breast cancer movement started back in, I guess it was the the late 80s. It was breast cancer advocates who, went to Washington. And that's how the DOD started distributing funds for breast cancer research. They heard our voices.
Thank you. Thank you. So for our next question, someone says, you spoke about a pro-inflammatory immune response that contributes to more aggressive cancers in Black women. Can anything be done before or during a diagnosis to mitigate the inflammation?
That's a hard one.
It's a very loaded question.
I don't think that anything is really known about that. Song, do you?
Yeah, I agree. I don't think we know like many anti-inflammation agents that can be used to improve the immunity against tumors. The immunotherapy drugs we are using, they are very specific to the targets, and they have also all kinds of side effects. For the prevention purpose, I think besides like keeping physically active, maintaining healthy weight and healthy diet, and lower your stress level, because stress can negatively impact your immune response. In addition to that, I don't know if we have good evidence on other preventive measures that we can use to improve immunity and to improve cancer outcomes. That's still a very active research area.
Thank you both. That was a really great question. And we have another question, a point of clarity. Someone wants clarity that what you had said, Black women from our ancestry have stronger immune cells, but have exhausted these cells. Is that what you were saying?
So
Yeah.
This is on a, we were looking at tumor cells from hundreds of people. So these things cannot be necessarily taken and applied to the individual level. It's just like, breast cancer risk factors when we know that, I'm trying to think of one that's kind of general. I don't know, being overweight increases risk of postmenopausal breast cancer. You can't really take that on a personal level. It's lots and lots of studies looking at, hundreds or thousands of people. So we had this finding, and this is a novel. This is first. We haven't really replicated it yet. That higher levels of immune cells in the tumors from Black women on average. And they tended to have this exhausted phenotype on average. And this is the first study to see this. Sorry for jumping in on you, Song.
Oh, no, not at all. That's beautifully answered.
Okay. Okay, so for our next question, in your talk, you mentioned breastfeeding and sufficient levels of vitamin D might impact outcomes for Black women with a breast cancer diagnosis. Are you recommending that all women, as much as possible, breastfeed and supplement with vitamin D?
That's a tough one too. I think that the data are strong enough to show that breastfeeding reduces risk of ER negative and triple negative breast cancer. The other thing is it's really good for babies to breastfeed them. It's good for their health. It creates a bond. It's good for brain development. So I think you can't go wrong with breastfeeding. But there's been a traditional lack of support for women to breastfeed. If you need to work, how does the workplace make that happen for you? Are you surrounded by people who can help you when, baby's not really doing well with nursing, or you need help? So it's good if you can be in an environment that makes it easier to breastfeed, because there can be a lot of impediments.
Okay, thank you. So for our next question, once diagnosed with triple-negative breast cancer, should Black women ask their doctors about specific treatment options, such as checkpoint inhibitors?
Again, we are not oncologists, so but my understanding on that is, for breast cancer treatment, most of the times it's fairly standardized, especially for the most common type of breast cancer, ER positive breast cancer. The common management is to get surgery and then maybe radiation therapy or adjuvant therapy with one type of hormonal drugs like tamoxifen or aromatase inhibitors. That would be enough, and that will help with a lot of patients. For triple-active breast cancer, which is more aggressive, and in 2019, FDA has approved triple-active breast cancer patients who are locally advanced or metastatic. So that means the cancer has spread out from breast, and immune checkpoint inhibitors can be used for that indication. So that's what FDA says.
Okay, great. These are really, really good questions.
Yes, they are.
Okay, so we have another question.
Sure.
You did speak about this in a previous answer. The question is about the exhausted state of the cells. Do we know what percentage of people have exhausted cells? And how do you know if your cells are exhausted? Is this something that you can, bring to your doctor and say, hey, my cells are exhausted? Or, how do we know? What do we know?
Yeah, I hope it will be as easy as that, but then the reality is always a little bit more complicated. For the exhaustion markers, there are a clinically approved measurement, such as PD-L1 expression, and that can be done before a doctor decides whether you are eligible for whether immune checkpoint inhibitor would be good for you or not. So using that test can help. Actually, the FDA approval for triple-active breast cancer requires patients to have PD-L1 expression positive. So I'm not sure exactly what's the percentage of patients have the exhausted immune cells as measured by PD-L1. But for individual patients, really that doesn't matter. It's as long as if you are PD-L1 positive and triple active and locally advanced, I think the immune checkpoint inhibitor is an option.
Okay, thank you. So for the next question, have these exhausted immune cells been discovered in other populations as well?
It is certainly, it's the exhaustion, T-cell exhaustion is a phenomenon that exists in not only breast cancer, but in many other cancers like melanoma, renal cancer, lung cancer. And actually the checkpoint inhibitors were developed specifically to target those exhausted immune cells, trying to revive them. So they can be reactivated to kill cancer cells. And then across cancer types and also across population of different reproductive backgrounds. So certainly in Black patients, in white patients, in Asian patients, Hispanic patients, some patients may all have that kind of phenotype. and can potentially benefit from the immunotherapy. It just at a population level, as Christine had mentioned earlier, when we look at the population level, on average, in our study, we found Black breast cancer patients tend to have higher likelihood to have the exhaustive immune phenotype.
Thank you. And for our next question, people want to know, how has the pandemic, if at all, impacted your research?
Thank you for that question. At first, well, we were all freaked out. At first, where we work, Roswell Park Comprehensive Cancer Center, sent everybody home. They kept some clinical activities going on, but the researchers, we left. But most of us, we don't work in the lab. We work on computers and we have data. So most of us just kept working remotely. By the middle of May, we were back because Roswell Park is, we're connected with the state and we're considered essential workers because we're a hospital. So we were all back. We got, socially distanced every room that we could get to. You have to swipe in and have your temp taken before you can get into work. So we feel very safe here, but it slowed things down. We were going to start that study with the New York State Cancer Registry, and they all got sent home. And so there were a lot of delays, but the National Cancer Institute and journals too have kind of given us a break. Okay, if you're late with stuff, it's all right. We understand this here, but boy, we all really have had to learn to adapt. And I miss having in-person meetings. I mean, this Zoom is great, but I miss seeing folks sitting around a table together.
I know, not the same. Dr. Yao, did you want to?
Yeah, I think in comparison to many other our colleagues at other places, we came back to office and back to kind of a normal work schedule fairly quickly. I think it's from March to May, just a few months. And then many of us are back to the office and we feel safe here. And but then, certainly some things, especially the patient enrollment has been slowed dramatically. And then things have been back up to the normal pace for many places, including here, so which is good.
Okay, wonderful, wonderful. Well, congratulations, before I forget, on receiving a grant on your research to continue your research. In your opinions, what is the best possible outcome that you would like to see for this research project?
Oh, yeah, that's a really great question, and I appreciate that. I think maybe I can start it, and then Christine can add to that. I think we are hoping to find out multiple things because this is a quite ambitious project that we designed. We want to know exactly whether Black patients have better response to immune checkpoint inhibitors. If they do, I think that gives us more justification to use immunotherapy in Black patients, because this study is not only for breast cancer, we include all types of cancer. As long as patients are eligible for immune checkpoint inhibitors, if we can show that Black patients indeed have better response to immune checkpoint inhibitors, then that can be used to bridge the gap in the survival between Black and white patients. And that's at the same time, because of the heightened immune response we often see in Black patients, we're also concerned whether they have more toxicities from the treatment. So we want to see whether the toxicity, whether there are differences in the Black-white patients in regards to toxicities after the treatment. And we certainly want to minimize that. And then the other major part of the research is led by my colleague, Charlie Kamen at University of Rochester, in trying to understand whether social, economic status and access to healthcare may hold some patients from getting treated with this new therapy, because it's the treatment is quite new and also quite expensive, and there are a lot of barriers to get access to this type of treatment. So we hope through our study we can provide some answers to that to help patients to get better access. Christine, do you want to?
No, I think you've said it all, and it's 2:00. I don't want to keep people hanging on, but we're so excited about it and really happy to be embarking on this.
Christine, are you sure? We want to make sure that I give you time to answer if you would like to, please.
Well, it's so gratifying that we started with this hypothesis about, immune response, and took that through the epidemiology studies and in healthy people, and then the differences in tumors, and that has led to this really big nationwide study that I think is really going to have an impact, I hope, on reducing disparities and gaps and access to the best treatment. That's it.
Thank you. Thank you. Well, I want to thank you both, Dr. Ambrosone and Dr. Yao, for such an informative program. And thank you to everybody for participating and having the best questions ever to ask both of them. I would like for everyone to please take a moment to fill out the survey at the end of the webinar. The survey will show up in the browser when the webinar ends, and the link will also be in the follow-up email. Thank you, everyone, and have a good day.
Thank you. Bye.